Good morning. My name is Heather, and I'm a research embryologist at Colorado Center for Reproductive Medicine. I'm excited to talk to you about the use of extracellular vesicles in reproduction because this is an area I've been involved in through my dissertation research. There's a growing interest in how EVs might be used to improve fertility, and could they one day be used to clinically to help support embryo development and improve fertility outcomes. So that's the big question. And today I'll share a brief summary of what I've worked on so far and how I plan to use the TSC-derived EVs in my research. So before I dive into the extracellular vesicles, let's first talk about one of the biggest challenges in reproductive medicine, and that's how aging affects fertility. This graph illustrates two key trends. As we age, the number of oocytes declines, and at the same time, we have a proportion of poor quality oocytes that increases. So this is a dual challenge that is a major factor in age-related fertility decline. We can't do much about the number of oocytes that decline, but maybe we can look at how to improve the quality of the remaining oocytes. And this is where maybe extracellular vesicles could be beneficial here. So let's take a closer look at what's happening inside the ovary as it ages. This diagram highlights four key factors that contribute to ovarian aging. The first being genetic abnormalities. So as eggs age, there's an increased risk of chromosomal errors, which then can result in aneuploidy blastocysts. And we do see an increase in this with our aged patients. Then there's impaired mitochondria. So mitochondria are the energy source of the cell, and as they become less efficient, the eggs lose energy needed for their normal function and development. There's also changes in antral fluid and immune response, and this is probably the area that I'm most interested in, looking at the changes of that follicular fluid, specifically with EVs, and how that changes over time. And then finally, with the altered extracellular matrix and vasculature. So we know that ovarian tissue, as it ages, it gets very stiff and becomes fibrotic, which can impact the ovulation of a healthy oocyte. To better understand how ovarian aging affects granulosa cells, and the granulosa cells are the supporting somatic cells within the follicle. So the diagram to the left, I'm specifically interested in the mural granulosa cells, which line the outer edge of the antrum and really contribute to a lot of the hormones, growth factors, proteins, and extracellular vesicles of that follicular fluid inside the follicle. So I conducted an RNA-seq study comparing young and aged pre-ovulatory mural granulosa cells. Through differential gene expression and differential microRNA expression, we identified a potential regulatory network of microRNAs that are targeting pathways involved in inflammation, angiogenesis, extracellular matrix remodeling, and inflammation recovery. So a lot of the same things we saw in the previous slide that really contribute to ovarian aging. One of the most striking findings was the evidence of an impaired immune response in the aged granulosa cells. Since ovulation is an inflammatory-like process, this dysfunction could contribute to ovulation failure, which tends to occur as the ovary ages. Also is the lack of proper inflammation recovery. So with ovulation every month, that inflammatory process is important to have inflammation recovery and go back to essentially baseline levels. Otherwise, this could lead to a chronic state of inflammation, which is often termed as inflammaging, which we see in a lot of aged tissues. So this is a persistent low-grade inflammation that can occur in aged tissues. So this highlights the molecular changes that occur in the aging granulosa cells, and it kind of opens the door for potential interventions and possibly using extracellular vesicles to help restore some of this function. So building on our understanding of ovarian aging, I also wanted to look at how extracellular vesicles in that follicular fluid and their cargo and how it changes with age and how it might be contributing to the decline in fertility. Specifically, I looked at differentially expressed microRNA between young and aged follicular fluid EVs. And you see a clear difference in how that cargo changes between young and aged. And the key highlight here is that dysregulated microRNAs from the aged follicular fluids target genes involved in pathways related to oxidative stress. So specifically the HIF1 pathway, which is critical in a cellular response to hypoxia, and it's also involved in follicular development and ovulation. Additionally, the age-range pathway, which is closely linked with oxidative stress and inflammation, both of which really contribute to ovarian aging. So given what we've seen with the impact of aging on the follicular fluid EVs and their role in oxidative stress, the next question is, can we intervene? So this brings me to how I want to use some of the TSC-derived EVs. You might be thinking it's a little strange talking about trophoblast stem cells in the ovary. It's not physiologically relevant, but we do know that with extracellular vesicles, regardless of their origin, they do have cargo like microRNA that can modulate various processes, including inflammation and oxidative stress response. So TSC EVs specifically have been shown to have beneficial properties and can influence cellular functions such as cellular senescence, which is very common in aged tissue. So by supplementing these TSC EVs into an in vitro mouse maturation model, we're hoping to see if these vesicles, despite not being physiologically native to the ovary, if they can still have a protective effect, improve maturation rates and reduce oxidative stress and support that mitochondrial function, which could hopefully eventually lead to good blastocyst development. And this could be a pilot study test on how EVs can be used in a clinical setting to improve oocyte quality and improve fertility outcomes. In addition to improving oocyte maturation, we're also looking at how TSC-derived EVs might affect the pre-implantation embryo. So we know in vitro embryo culture can be a stressful environment for the developing embryo. So as much as scientists and embryologists have worked to optimize the in vitro culture, it still doesn't match a healthy in vivo environment. So we're hoping to see if some of these trophoblast stem cell derived EVs can help mitigate some of that stress and help prime the embryo for implantation. So speaking of implantation, I'm switching gears a little bit and talking about how we can model the peri-implantation stage embryo. This is a crucial stage in early pregnancy where the blastocyst is just beginning to differentiate and prepare for implantation into the uterine lining. In this diagram, you can see how we can use the human embryo in culture then past the blastocyst stage. And we've used this model for several studies to measure the implantation potential of an embryo. This allows us to study the differentiation of trophoblast cells, which is key to successful implantation. And we can investigate how the different trophoblast populations are developing and how they might be influenced by different factors such as extracellular vesicles. So I have a video here so that you can see what that looks like. So we have a human embryo here and it attaches by day 8, and you can see the progression of the outgrowth with every day. By day 10, we start to see these big syncytial plaques, which are nice, and I have a photo later that shows that they stain positive for HCG. And as it continues to develop between days 10 and day 12, you see a lot of cells that break away from the embryo. We call these migratory trophoblast cells, so they're looking for that maternal blood supply. So this video highlights the differentiation and migration of the trophoblast population, which is key to establishing that early pregnancy, and it can be used as a good model to study this very early stage of embryo development. Additionally, we have a novel approach to modeling peri-implantation. We have, in collaboration with a lab at UT Southwestern, we've established a protocol for generating these blastocyst-like structures, these blastoids, that are known to mimic many aspects of the natural blastocyst development. They form a fluid-filled like cavity, much like the blastocyst, and they contain all 3 cell lineages. And we've shown that they behave very similarly to blastocysts when cultured in vitro for extended periods of time. So blastoids offer us a unique advantage. They allow us to study early implantation and trophoblast differentiation in a reproducible model. So unlike human embryos, which are limited in number with high heterogeneity, blastoids can be produced in large quantities. and come from the same cell line, so have the same genetic background, making them potentially more ideal to study how EVs can improve implantation. I have another video here. So here's a video of a blastoid in an extended culture. So it attaches to the dish much like the blastocyst did in the human embryo. And you can observe the progression of the outgrowth with the syncytialization. And we also begin to see those migratory cells breaking away from the blastoid. So we've always looked for ways to improve our in vitro culture conditions for this peri-implantation stage embryo. So what we did was co-cultured some blastocysts with human endometrial stromal cells that were obtained through endometrial biopsies with consent from donors. And we've seen a benefit to adding these supporting cells to the in vitro culture for our extended culture model. We've also cultured blastocysts with the conditioned media from those endometrial stromal cells. And as you can see on the graph on the right, we did see a marked improvement in that outgrowth area for the blastocysts that were cultured in conditioned media compared to those that were cultured either alone on fibronectin-coated plates or with the endometrial stromal cells. So this begs the question, could it be the extracellular vesicles in the stromal cell conditioned media that is aiding in this implantation improvement that we see? So that's something that we're still actively investigating. It's part of the reason we're interested in exploring the TSC-derived EVs to see if we can improve implantation. So with that said, we're planning to extend our studies to investigate the role of the TSC-derived EVs using the embryo and blastoid extended culture models, hoping that we can use these models to explore how EVs can enhance that trophoblast differentiation, which is critical for implantation and placental development. So to summarize, I've explored the potential use of the TSCs in enhancing reproductive outcomes, specifically in the context of ovarian aging and early embryo development. We've seen how changes in EV cargo, especially the microRNAs, could be impacting fertility, and I'm excited to see if the TSC-derived EVs can be used to improve oocyte quality, embryo development, and the implantation potential. So with that, I'd like to thank you all for your time and attention and see if you have any questions or would like to open this up to discussion.